Good morning, student. So this is our unit 4 plant physiology in chapter 13 photosynthesis in higher plants. Today we'll study how plants do photosynthesis. So we all know that all animals including human beings depend on plants for their food. Have you ever wondered from where plants get their food? Green plants in fact have to make rather synthesize the food they need, and all other organisms depend on them for their needs. Green plants carry out photosynthesis. It's a physiochemical process and in which they use light energy to drive the synthesis of organic compounds. So ultimately, all living organisms on earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on earth. So photosynthesis is important due to two reasons. It is the primary source of all food on earth. It's also responsible for the release of oxygen into the atmosphere by green plants. So what will happen if plants are not photosynthesizing? So in this chapter, we will learn about all the processes which are involved in the process of photosynthesis. So photosynthesis is an enzyme regulated anabolic process. Anabolic means synthesizing process of manufacturing of organic compounds inside the chlorophyll containing cells from carbon dioxide and water with the help of sunlight as a source of energy. Photosynthesis is a chemical process, uses light energy to synthesize organic compounds. And importance of photosynthesis includes that it gives us primary source of food and it releases oxygen to atmosphere. In 10th class, we have studied about the experiments to look for starch formation in two types of leaves, variegated leaf or a leaf that was partially covered with black paper and one that was exposed to light. So these experiments we all have done in the previous class. So now we'll do some more experiments that Joseph Priestley in 1970 performed a series of experiments that revealed the essential role of air in growth of green plants. Priestley, you may recall, discovered oxygen in 1774. So Priestley observed that a candle burning in a closed space, a bell jar, soon gets extinguished. Similarly, a mouse would soon suffocate in the closed space. He concluded that a burning candle or an animal that breathed the air both somehow damage the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plants restore to the air whatever breathing animals and burning candles remove. In these diagrams, you can see A and B are showing that in A, mouse is alive and candle is burning. Both are giving out carbon dioxide and the candle will extinguish and mouse will die. But in next C and D, you can see when we have placed a mint plant in the bell jar, then you can see that all the three are alive. How it can happen that candle needs oxygen for burning and it gets from mint plant, and same as mouse also needs oxygen, that is mint plant is giving, and they both are giving carbon dioxide, and mint plant needs carbon dioxide for photosynthesis. So all the three are alive. Jan Eganhouse showed that sunlight is essential to plant process that somehow purifies the air, followed by burning candles or breathing animals. Eganhouse, in an elegant experiment with an aquatic plant, showed that in bright sunlight, small bubbles are formed around the green plants, while in the dark they didn't. You can see in this diagram that some hydrilla plant is kept under the funnel and bubbles are giving out. Later he identified these bubbles to be of oxygen. Hence he showed that it's only the green part of plants that could release oxygen. In 1854, Julius von Sasch proved that plants produce glucose while growing and stores them as starch. He explained that plants have green substance which is present within a cell in specialized bodies. He found that the green parts in plant is where glucose is made and that the glucose is usually stored as starch. So T.W. Engelmann showed that split light using prism Vibgyor, you all know Vibgyor, green algae Cladophora placed in a suspension of aerobic bacteria. Bacteria were used to detect the sites of oxygen evolution. Means bacteria will gather, aerobic bacteria will gather around the oxygen evolutionary part. He observed that the bacteria accumulated mainly in the region of blue and red light of split spectrum. It explained the absorption spectra of chlorophyll A and B. By the middle of the 19th century, the key features of plant photosynthesis were known, namely that plants could use light energy to make carbohydrates from carbon dioxide and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organisms was thus understood as carbon dioxide plus water in the presence of light going to make carbohydrate and plus oxygen, where CH2O represents as a carbohydrate. It may be glucose, a 6 carbon molecule. Cornelius Van Niel, he did experiment with purple and green bacteria and demonstrated photosynthesis is a light-dependent process. He explained hydrogen from water reduces carbon dioxide to carbohydrates. He conducted that oxygen comes from water and not from carbon dioxide. Finally, the correct equation for photosynthesis was discovered that 6 molecules of carbon dioxide when react with 12 molecules of water, they're going to make 1 molecule of glucose plus 6 molecules of water and 6 molecules of oxygens are evolved.